And I want to welcome everyone. I'm Sharon Terry, the president and CEO of PXE International. Delighted to have this global meeting spread over many months, actually the whole year. We have talks all the way out to December so that everybody can hear any talk they want to, either live like today or on recording on PXE International's YouTube station. And so without further ado, I'm going to introduce Olivier Le Saux, not with any formal introduction because I know him for a very long time. And we have collaborated for a very long time. I have hanging on my wall here a special photograph diagram. I'm not sure what word to use that would make sense to lay folks of the gene discovery that he gave to me. And it's very special. I see it every day. And I'm very happy to have collaborated all these years, which are in excess of 20 now. And so without further ado, I'm turning it over to you, Olivier. Thank you.
Well, thank you for this introductions. I'm glad you still have that.
Yes, I don't. Someone asked me if they could put it in a museum in Germany for a little while and exhibit on genomics and patient power and blah, blah, blah. And I told them no, even though they said they didn't share it for $1 million. I said no.
Well, if anything happens to it, I have another one on my desk.
Okay, good. Well, we each need one, so.
Because those things are, I had to make them in duplicate. So one just to look at it, and another one to cut the pens off and do some more analysis on it. So this is why there's 2 copies of everything. I'm going to try to share my screen and begin with my presentation. Again, that's this one, yes.
And then we'll remind you, some people are low vision, and so they'll count on your descriptions since they won't be able to see the slides very well.
Yes, and by any means, interrupt me because I'm not necessarily going to describe everything in super details just by the force of nature. I when I go down in my presentation and I am in my presentation mode, and that will necessarily stop or make things very clear because it's obvious to me. It's not necessarily obvious to you, so don't hesitate to interrupt me so that I can go back and explain what you have not understood, or I didn't make clear enough.
Great, thank you.
All right, so I can play this. Okay, everybody sees this?
Yes.
All right, so what I'm going to explain here today is really the evolution of the work that has taken, I'll say, 2 decades. So this is not something that happens yesterday or even in the last year. This is really an idea that's evolved over time into what it is today. And it's, I think it's a relatively significant discovery that we made because it will probably have impact on how we understand PXE and eventually how we treat PXE. So without getting too much into the details of the title, it's bone marrow-derived ABCC6, which is the expression of ABCC6 in bone marrow, is really an important determinant of how the calcification evolves and how the pathology, the disease evolves. So I want to take you, and it's a little like heavy on data, this presentation. So again, if something is not clear, make sure that you I have 2 screens here in my presentation. So the other, your video feed is on the other side. So say something so that I can actually see you and interrupt and go back and to explain, see if you need to.
Okay, that sounds fine. I think we'll have you explain what you wish, and then we'll open to questions at the end.
Okay, so on the top of the slide here, I have the molecular pathway that, as we understand it now, is really important in determining the calcification in PXE. So it starts out with ABCC6, and the next protein, it's actually an enzyme after this that is really important, is ENPP1. But the story doesn't stop here. You need to have another one down the road. It's called CD73, and another important one called TNAP here for tissue non-specific alkaline phosphatase. It's an alkaline phosphatase that removes phosphate from things, and I'll explain in a minute what this is all about. So I kind of dubbed this the ABC6 pathway with all these 4 elements that are critical for the development of calcification in PXE when things are not going according to plan. So really the story starts in the liver, where the majority of ABCC6 is expressed. And the function of ABCC6, as we understand it now, is to pump out the cells ATP, which is an energy molecules that the cells normally need to use to drive all sort of function. The function of ATP is really to release this ATP, a small amount of ATP outside, and that gets into the blood vessels of the liver. That ATP is quickly degraded by the secondary enzyme, the ENPP1 enzyme. and will remove 2 of the phosphates of this ATP and generates pyrophosphates. And by now, I think you know that pyrophosphate is a key element that prevents calcification, and that when there isn't enough, PXE arises. So in this figure, we have ABCC6 releasing ATP in liver vasculature and liver blood vessels. And this ATP is very, very quickly, within milliseconds, it's actually degraded by ENPP1, generating this pyrophosphate here, which eventually gets into the bloodstream, is distributed throughout the body, and normally prevents abnormal calcification. So I'm showing kidneys, the eyes, blood vessels, and the skin here for PXE, but there are also a few other tissues that are affected. So that's a general principle, but the story completely doesn't really stop here, as I mentioned, because just below on this figure here, there's another molecule called AMP, which is the byproduct of ENPP1 activity. And this adenosine monophosphate is further degraded by this CD73 enzyme, generating adenosine. You probably heard of that term, adenosine, and it does a lot of things. It has a lot of regulatory function in the body, and one of them is to stop TNAP from functioning. And I put a stop sign over TNAP because TNAP does many things, and among other things, it will be happily degrading pyrophosphates if given a chance. So you can see that this whole pathway produces ATP. From there, we produce this pyrophosphate that stops calcification. But further down the line in this pathway, we see that the adenosine here makes sure that this pyrophosphate stays intact and is not being degraded by stopping the expression and the activity of TNAP. So we have a good pathway. We produce something and we make sure it's not degraded. And that, in normal circumstances, this is where abnormal calcification is somewhat prevented. Otherwise, I mentioned TNAP would happily degrade pyrophosphate, and I'll show you some elements of this and some proof of this. An important aspect of this presentation is associated with the fact that ABCC6 is mostly expressed in the liver. I exaggerated the color here and the size of the font to show that the bulk of the expression is found in hepatocytes, which are the main cells of the liver. And the next tissue that expresses ABCC6 to a detectable and good level are the kidneys. And the kidneys represent about, if we do some rough comparison, it's about 5% of what you found in the liver. So really the liver is the heavy weight here in terms of ABCC6 expression. There is some detectable expression in the intestines and a few other cells, but in mass and volume, it's really the liver, which is the king here. And this, over the year, has given rise to a perception that the liver is absolutely essential in determining calcification and also how PXE develops. And by the natural follow-up on things, it's, because it's mostly expressed there, most studies have actually been done on ABCC6 in the liver. I'm citing a few papers here, a few publications. There are many more. And the articles and research has been done on expression of ABCC6 in other tissues are few and far in between. There's just a handful of them altogether. Some focusing on the kidneys, some focusing on tissue with elastic fibers, which are calcified in PXE, but not a whole lot. And this has given rise again to this sort of urban legend that the failure of the liver to produce an inhibitor of calcification, pyrophosphate, is sufficient to cause PXE altogether. However, there was really very limited direct evidence that this disease model is actually real. It's, as I said, it's more of an urban legend altogether, and an accepted notions that is perpetuated in publications and in conferences and meetings. But there is not a whole lot of things actually supported this. It's very much like, I hope I'm not going to get in trouble by saying this. It's like, using fish oil is good for your health and your vasculature. Well, there's not a whole lot of scientific evidence behind that either. There's a lot of things like this that we humans very quickly take for granted because everybody's repeating the same thing. And there was a little bit of that in respect to the liver and PXE. However, ABCC6 expression in other tissue immune cells have been somewhat neglected over time. And I have to say that I had an interest in looking somewhere else than the liver because I were part of some of the studies in 2003, 2006, and 2011 that we specifically documented the expression of ABCC6 in blood cells, so in white cells, and even in red cells. So that was surprising, and we were wondering why, and that didn't go anywhere because we were busy studying things associated with the liver, but not so much with this. So I'm just guilty as others on that front. But still, it was something that I can never completely forget. It was in the background of my head. So I mentioned this liver hypothesis as being important, but there is also another school of thought in the field of PXE and research, what we call it the local cell hypothesis. And essentially, there were numbers of investigators that were really focusing and working on I don't want to say proving, but they were working on the fact that the cells that produce elastic fibers that are calcified in PXE, notably fibroblasts, type of cells in the skin that make the elastic fiber. And these cells, they claim, play a major role in the calcification of PXE. And there are, of course, a number of papers, but nothing really like a smoking gun. It was mostly circumstantial evidence in there, but and one argument that really run against this specifically is that there is no ABCC expression to speak of in fibroblasts. It's detectable, but it's a trickle, to say the least. So this school of thought and this hypothesis had a few proponents, and the majority, I think, of researchers on PXE really didn't think much of it. But it had the merits to exist because so far, as I mentioned, the liver hypothesis that everybody believed in, well, we didn't have really hard evidence to support that. So that hypothesis was just as good as the other one. Until about 2017, when this paper came out, there were a few controversial elements of that paper, but they really approached the research in a slightly different angle. Theirs were not necessarily people, Harry Dietz, Hal Dietz at the end of the author list here, and Shira Ziegler, really approach this idea. They're coming from outside the PXE field. They are Marfan syndrome people, and they came with a slightly different outlook on how to approach this problem. And what they essentially suggested at the end of their research and these experiments is that liver might not be as important as one thought, and the contribution of other tissues might be non-negligible, and particularly the role of TNAP. If you remember, I mentioned TNAP a few moments ago. Well, let's get back to it. I'll tell you what TNAP is doing here. So in the case of PXE, we don't have ABCC expression in the liver and other tissues. Therefore, there's a lot less ATP that is being effluxed out of the cell or pumped out of the cell into the blood vessels of the liver. So in this whole molecular pathway that I mentioned before, if there is less ATP, well, the following enzyme, ENPP1, cannot make as much pyrophosphate from it. So there's a lot less. So on my picture here, I describe a smaller pyrophosphate. If there's less pyrophosphate that gets into the blood circulation, well, there's a lot more abnormal calcification that happens, and that's PXE. But as I mentioned, there is something that happens after this ENPP1 enzyme. If there is less pyrophosphate, there's also less adenosine monophosphate, AMP, on this figure. That means there is less adenosine down the road also, because CD73 cannot make as much from the other things. It's just, it's like a snowballing effect. It's just getting smaller in that particular case. And that stop signs over TNAP is somewhat removed here. That means if there's less adenosine, it allows TNAPs to be a lot more expressed. And if TNAP is more expressed, well, it's going to be happily degrading pyrophosphate. If you recall earlier on, I said this pathway produces PPI and make sure it's not degraded. Well, if you don't have ATP pumped out by ABCC6, well, there's a lot less PPI, but there's also a lot more TNAP, this time degrading happily pyrophosphate. So it really aggravates the situation in that sense. And what these people thought is, man, perhaps TNAP could be a way for us to reduce calcification, and I kind of investigate this. And so they use a drug called SBI 425, which is an anti-TNAP drug, and they treated mice, PXE mice, for this. And I'm going to try to explain that figure here. You're looking at the blocks that are blue, red, and yellow here. And they've used a regular ABCC6 mice. They treated it with something like a placebo. And so here's the SBI 425 here. This is the red block. This is where they treated the mice with. So when they actually treated the mice with a placebo, they have normal calcification in these mice. And it's at the blue box that you see here. And then when they're treating the mice with the drug, this anti-TNAP drug, you can see there's a significant reduction in calcification outcome. And then they use a control drug, etidronate, which is a biphosphonate that has been used in clinical trial in PXE with some success. We know it's suppressed calcification. And so they're using it as a comparison marker here. And there is really a significant decrease in the calcification outcome in the mice when they use this anti-TNAP drug. So this was really interesting. And TNAP is not expressed in the liver, it's expressed everywhere else. So it's showing us that maybe the liver alone is not really sufficient. But they've really pushed this further, and I'm going to explain that in a second. The other thing they've noticed as well is when you treat whether wild-type mouse or ABCC6 mice with this drug, anti-TNAP drug, they found that there was no significant change in the pyphosphate levels in the blood of the wild-type mouse or the ABCC6 knockout, the PXE knockout mice, as you can see here. And this was relatively interesting, and I'll come back to this in a moment. But what they've done in this complicated figure here, I just want you to focus on the yellow and blue boxes and the green boxes on the bottom of the figure here. If they've used genetic techniques where instead of removing ABCC6 from the entire mouse, they only removed ABCC6 from the liver. So it's expressed everywhere else, but not in the liver. So we call this a tissue-specific knockout. And so the figure is complicated here. They look at multiple things, and they're looking at 20 weeks of age, which is roughly 5 months of age in the mice development, and also at 1 year of age. 5 months of age, it's an adult mouse, well-developed. Mouse in a cage can live up to 3 years. So you can see 1 year of age in a mouse, it's about a third of its life. It's a significant amount of time for a mouse life. So this is why they use 5 months and 1 year. It's two important components in the life development and their life cycle. And so in the blue boxes here, this is a normal PXE mouse, no ABCC6 expression at all in the mouse. And this is the normal level of calcification one would get at 5 months of age. And when they knocked out ABCC6 specifically in the liver, so no expression in the liver, but expression everywhere else, these mice simply don't develop calcification at 5 months of age. It's not detectable. It's down at the bottom here. And they let the mice age further to 1 year of age. Here's the arrow pointing out down. And here is a control mouse. Lots of calcification, lots of PXE calcifications. And the same one that doesn't have ABCC6 expression in the liver only starts to show calcification. So there's a significant delay in development of the disease and the calcification when it's not expressed in the liver. And that really means an interpretation is the liver is not the only actor or a tissue that regulates calcification in PXE. There are others as well. And the work with TNAP is also showing the same thing. So this was really a significant, the first significant elements that would indicate perhaps the liver is not the only player in town. The story doesn't stop here. I'm trying to show it you chronologically, or not chronologically as it happens, because now I'm going to have to take you back 20 years or so when one of my graduate students who came here from Germany to work on his master, I was explaining what PXE is to him, and one of his remarks was like, oh, look, the PXE manifestations really match the main lymph nodes that we see under the skin. So in the figure here, this is a simple representation of where the lymph node concentrations are in the body, and they're around the neck. They're also around the axilla. The axillary lymph nodes are specifically where we see PXE symptoms, and also in the groin area. And if you go further, it's in the the fold of the arms or behind the knees as well. And so he made that observation that there is a good match between the lymph nodes under the skin and the PXE manifestation. And of course, I had to dismiss promptly his idea and said, oh, come on, there's no inflammation in PXE. Just forget about it. He forgot about it, but I did not. I'm putting here a lymph nodes to show you what these things are doing. This is essentially an immune checkpoint in the body. It's a very small kidney shape organs that drains lymphatic fluid from the entire body into those checkpoints here. And they have also blood vessels coming in, bringing immune cells so that there is an immune checking, making sure there's nothing wrong that floats around the body, like infections, bacteria, and other foreign bodies. So they're really there at strategic places in the body to provide some immune checkpoints and making sure everything is fine. This is why when you go to doctors, they touch here to make sure you don't have any problems with lymph nodes. If they are swollen, it tends to indicate that there is something wrong going on. So even if I dismissed the idea from my graduate students all these years ago, as I said, I did not quite forget what he said. And the idea reemerged in several instances of women, one in particular, many, many years later, when I had a phone call with Dr. Elaine Davies, who's Canadian. She was working, she might be retired by now. She was working at McGill's in Montreal. And I had this phone conversation with her, and we were talking about lymphatic fluids, skin. She's an elastic fiber person. And the idea reemerged, and yeah, maybe there's something related to lymphatic fluid or lymphatic vessels and maybe lymph nodes as well. So with this reemerging again, I had to start investigating this possibility. And so here's the figure that shows the correlation between lymph nodes under the skin and the skin manifestations in PXE. And I had to find the corresponding lymph nodes in mice. So I put the mice down, I look at what they are and what the correspondences are, and I had to go get those lymph nodes in mice. And lo and behold, what I found was something I did not expect, and that often happens in science. You follow up an idea and you found things that are unexpected. So serendipity is a significant component of our research. And on the top left corner here, this is a circular lymph node that sits right under the skin of the mouse. This is the inguinal on the belly of the mice. And sure enough, I did find, I don't know if you can see my arrow moving, but I did find calcifications indicated by this little arrow here and these black dots. They're supposed to be red staining, but this picture it shows as a dark brown, almost black. I found mice that had calcification within the lymph nodes, but I found a lot of calcifications in the tissue surrounding the lymph nodes. And in lymph nodes, they're also in the lumbar area, that sits right next to the aorta, major blood vessel there. And I found lots of calcifications in the wall of this aorta blood vessels that are shown in these pictures here, and lots of all little calcification in tissues within the immediate surrounding of the lymph nodes, but nowhere else. So this was really in the first indication that perhaps there was really something related to the lymph nodes. The other things I've done in this series of experiment and exploration is looking at the whiskers. So you probably know that one of the defining characteristics of the PXE mice is where the calcification in its mice, and it's in their whiskers. So the whiskers are tactile hair. Your cat has whiskers, and many other animals as well. We don't. But these are interesting tissues because they are tactile. That means they can sense something. They're attached to nerve endings in there, and they have a big bulb at the bottom of the hair shaft. And this bulb is vascularized. There's blood vessels in it, and you can easily see blood. When you cut the tissues and look through histology, you can actually see blood remnants in there. And this is the capsule, the bulb that calcifies in the mice. And one thing I actually did look in there is for the presence of lymphatic vessels. So these lymphatic vessels are not like the blood vessels. They are draining this little fluid that bath your tissues and your cells, all of them. And so they are really important elements, as I mentioned earlier, in the immune checkpoints. Right? And they will be draining whatever bacteria and other foreign bodies into the body that enters the body to the lymph nodes. That's really their primary function. And so I looked at those lymphatic vessels in the whiskers in the mice, and lo and behold, I found lots of them. It's actually what's shown in green on this image. There are lots and lots of lymphatic vessels in the whiskers that heavily calcify in the mice. So this was another indication, oh, this is interesting. I raised an eyebrow. When I started to look at the literature, there is nothing at all that described lymphatic vessels in the whiskers before. So this was actually a discovery. It's the first time that's been observed that there are a lot of lymphatic vessels there. Not only there's blood, but there's also lymphatic vessels. One thing leading to another, we've discovered also what is called lymphangiogenesis. It's a mouthful, but it means it's the growth of lymphatic vessels in the PXE mice, but also in PXE patient samples skin that I obtained from my French collaborators in Angers in France. And these are the bars. This graph is showing the black bars. If you compare them to the white bars, which are the wild-type mice, you can see there's a lot more. Those bars are bigger than the white bars. And that really indicates that in the two different areas of the skin of the mice, I see a lot more lymphatic vessels. And in the skin biopsies from PXE patients, also find a significant increase in lymphatic vessels. So it's not something we all see in the mice. It seems to be happening in human as well. Why is this important, this lymphangiogenesis? It's because it's tied up to inflammation. You can see inflammation does a lot of things. Injury or infections leads to inflammations. Exercise can lead to good inflammation. You've heard that there's good and bad inflammation. But one thing that is tied up to inflammation very closely is this lymphangiogenesis. If there's something that goes wrong, the body is essentially creating more lymphatic vessels to increase the drainage and to improve the inflammation process and therefore the clearance of whatever foreign body or infection occurs. So this is another clue that there is maybe something related to lymphatic vessels, lymph node, and ultimately inflammation in PXE. So you can see it's a slowly evolving idea It's taken a lot of years and slowly also a few more minds to put together coming down to this idea. And in 2018, there was this publication suggesting that there is maybe a low-grade inflammation that exists in PXE patients, and perhaps in the form of flares that happening every once in a while, and that tends to be preceding an expansion of the calcifications in the skin or the skin lesions in patients. It seems perhaps to be mostly visible so far in teenagers rather than older adults. And how really do we try to demonstrate that inflammation is there, or that ABCC6 in immune cells driving inflammations are important in calcification developments? Is, well, we kind of use a sledgehammer approach here that we can relatively easily do with mice, which is a bone marrow transplant. So all your blood cells, including the white cells that drives immunity, are derived from the bone marrow. So if one replaced the bone marrow in a PXE knockout mouse, then perhaps we can see something different when it comes to calcification in the mice, and this is what we've done. So it's a complicated figure here, but we did a bone marrow transplant at roughly 2 months of age and let the mice age up to about 6 months. Again, 5-6 months, it's the same timeline that you've seen in your other publication. And then we kill the mice and harvest the tissues to look at what's going on. Again, why 6 months? Is because when we look at the calcification developments in the whiskers in those animals, 6 months is a good middle time point when the calcification is the most active in these animals. And we did 3 things. In science, we do experiments, but we do a lot of controls. We have to demonstrate that the experiments are real, that the results can really be solidly interpreted and believed in. So in order to do this, one has to do controls. So we did controls that are depicted here in blue in particular, where we did a wild-type bone marrow transplant into a wild-type mouse. So just to make sure that the bone marrow process and procedures is not affecting calcification in itself. We've done also a knockout on knockouts on a PXE mouse on a PXE mouse. It's just a converse, so we have to do those controls in order to, as I said, to ascertain the results. And the key component of the experiments is we take a wild-type bone marrow and put it into a knockout mouse, or we do the reverse. We take a knockout bone marrow and put it into a wild-type mouse. And then we do a number of verifications. We looked at the phenotypes, so that means we look at fibrous calcifications, we look at heart calcifications, kidney calcification, we determine pyrophosphate, we looked at ABCC6 expressions in the bone marrow, in the leukocytes, and white cells, and a bunch of other controls. So it's been a significant undertaking altogether and doing all of this, but we have to cross all the T's and dot all the I's here. And again, complicated figure, lots of data here. You can see down at the bottom of this figure, the blue controls and the green experimental data. And what I want you simply to focus is on one thing that are indicated by those red arrows here, this one bar here in the fibrosate, in the kidneys, and the heart calcification. And if you compare that bar to the black bar, which is our baseline control here, we found that when we put a wild-type bone marrow into a PXE mouse, so we reconstitute ABCC6 expression in the bone marrow, we have nearly 40% less calcification in the whiskers, 30% less in the kidneys, and 60% less in the heart calcification, which is an inducible phenotype in the mice. It's a very useful marker that the heart calcification that we can use in the mice. So we can see it's pretty consistent across all these 3 tissues here that we have a lot less calcifications when we have normal bone marrow, wild-type bone marrow, into a PXE mouse. When we do the opposite, when we put a knockout marrow into the wild-type mouse, it does absolutely nothing. There's no effect whatsoever. But what we see here is wild-type marrow will reduce calcification. And this is very important because it's telling us that immune cells or red cells, white cells in the blood deriving from the bone marrow seems to be contributing to calcification inhibition. And so put this into the perspective with what's happening in the liver as well. So it looks like it's a significant contribution of the calcification inhibition is coming from the bone marrow and therefore the blood cells. The other interesting thing that we observed here, it's again this blue arrow here, when we reconstituted wild-type marrow into the PXE mouse, we have less calcification. The plasma pyrophosphate, or the pyrophosphate in the blood of these mice didn't change whatsoever. We observed exactly the same amount level, So we didn't change pyrophosphate levels, and yet we changed calcification. It's the same thing that the Ziegler paper was describing before with their TNAP inhibitions. It prevented calcification, but it didn't really change pyrophosphate levels in the plasma. And of course, we had to do some more controls. We had to take the tissues, section it, and look at specifically at calcification. So we can skip that part of the figure here. So how can calcification inhibition be restored in the Ziegler paper and this story here without any change in blood pyrophosphate? Because so far it's really been the idea that pyrophosphate in the blood was really the marker that tells or the susceptibility for calcification. Well, we've seen this before. It's not an absolute marker or reference. These are two different mice, the regular genetically engineered ABCC6 knockout, and another one called C3H, which is a naturally occurring ABCC6 knockout. And at 12 months of age, they have exactly the same levels of pyrophosphate levels in their blood, and they have a vastly different amount of calcification in their whiskers. It's a little small on that figure, but the straight engineered ABCC6 knockouts have lots of calcifications, and the naturally occurring mutation in the C3H mice have barely any calcification at the same age. Yet they have the same plasma levels in their blood. And so we've suspected for some time that pyrophosphate in the blood was not really a good match for calcification severity or susceptibility in PXE. It's an important component, but it does not quite reflect what's happening in the actual tissues where the elastic fiber calcification happens. So patients from the same family can have very different severity, and if they are from the same family, they have the same mutations and they have the same, likely the same pyrophosphate levels. We know that PXE versus generalized arterial calcification of infancy, which can be also coming from ABCC6 mutations, which is a very, very severe form of PXE. They have the same mutations, very different phenotypes. You know, GACI is a lot more severe than PXE, and yet they have the same amount of pyrophosphate, give or take. So there are a lot of indications over the time where we see a mismatch between blood pyrophosphate and calcification outcome. And going back to the inflammation, we've been working closely with the French PXE patients and Dr. Martin in France. And it's been some time since we found evidence of inflammation in PXE patients. On those images, you can see a typical redness of the skin that happens in the neck or the axilla on those patients. They tend to describe that they have those flare red inflammations before the lesions expand in sizes. And by looking into their blood, we found also evidence of inflammation markers present there. It's called interleukin-6 or CCL5. These are really evidence of inflammations occurring in the patients. And recently, they, not me, managed to find a PXE patient, I think 13-year-old boy, who arrived at the consultation center in Angers, France, right in the middle of a flare of inflammation. So they were able to get a skin biopsy from him, from blood, and they've done some sophisticated molecular biology analysis, which is known that tables with various colors, red, blue, and greens, down here showing the expressions, showing infiltration of inflammation cells in the skin right where the lesions are, and also the expressions and various genes being activated that really corresponds to inflammation. So this is a new perspective now that we're seeing in PXE, that inflammation seems to be contributing to calcification, and inflammation means immune cells. So that means ABCC6 and immune cells are very important in determining the calcification outcome alongside with the liver. So what we found, and that's the summary of my presentation, is that restoring a normal ABCC6 functions in the bone marrow and therefore in immune cells significantly improve calcification outcome. I can tell you right off the bat that bone marrow is not a good approach to cure PXE. I can tell you this from first-hand experience. Bone marrow is not a good approach for anything, but there could be other ways to address inflammation. So if we're using the vibrissae and the whiskers in the mice as a measure of PXE in these animals, really our data suggests that immune cells account for nearly 40% of the calcification potentials that exist in the body. And the rest is probably coming from the liver and perhaps other tissue as well. And we have to figure that out, whether any other things besides the liver is important. So my results and that of my colleagues also supports the notion that the liver alone is necessary, but it's absolutely not sufficient to repress calcification. And that suggests a cooperation between the liver and immune cells in the regulation of calcification. Regulation of calcification via ABCC6 also requires assistance from other local and systemic modulators. And in that particular case, I'm talking about immune cells, but also the process of inflammation seems to be an important component in the development of calcification. And that does not necessarily affect pyrophosphate levels in the blood. And this is very important when it comes to the clinic. Can we use pyrophosphate levels in the blood to determine the susceptibility of calcification and the outcome and perhaps a treatment effect? The answer is probably no, or at least we have to use this with a grain of salt. So these two important components, inflammation and pyrophosphate levels, is something that needs to be taken into account for the future, I think, when it comes to potential treatment. So targeting pyrophosphate in the blood, yes, probably not enough. Should we look at calcification? No, sorry. So we look at inflammation as an important component for treatments in the future? Yes, possibly. This needs to be confirmed, and there are a lot more investigation that needs to happen. But it looks like flare of inflammation has really contributed to the calcification of PXE, and eventually will probably have to be taken into account for future treatments. I hope I didn't bore you too much with too much specific data. Lots of people were involved over the years. As I mentioned, this is really a study that spans well over 2 decades. And it's come to fruition finally with a publication that just came out. And that's really opened another venues for research and investigation. So we have people from France, people from my lab at University of Hawaii, but also collaborated with Koen van de Wetering in Philadelphia, and also the Hungarian crowd with András Várgyi and others, and particularly Viola Pomodi, who came to my lab for numbers of years and contributed significantly to all these experiments that I showed you. All right, and I'll stop here and I'll be happy to take your questions.
All right, thank you very much, Olivier. Can you take down your slide because it helps if people can see you? Okay. And so you can ask questions by typing them in the chat. You can raise your hand with the Zoom hand, or you can raise your real hand, or you can unmute. Lots of options.
I have a quick question. Thank you so much. That was really helpful and stretching some of my knowledge on this. So I think you were kind of talking about this. So in your view, does it help to limit calcification by limiting inflammation through one means or another?
I would say yes, tentatively yes. This is what all these experiments are showing, that inflammation is a significant contributor to calcification. So if that's the case indeed, yes, I would think moderating inflammation would probably be an important component of a treatment in the future. We'll have to see what works well and the best, but I wouldn't be surprised if this becomes an important aspect of treatment in the future.
And just one quick follow-up to your early points about kind of quote good inflammation and bad inflammation. As far as you know, would there be any kind of difference in the type of inflammation or the cause or etc.?
At this point, I don't know. I really don't know if it's a good or bad inflammations that contribute to calcifications. I would say by default, I would say it's probably the bad inflammations that is contributing to further calcification. But I can't say at this point yet. We don't have enough information on that. So.
Great. Thank you.
Quick question.
Hang on a second, Bob. First, we have Mark, then Colleen, then you. So Mark, you're on.
Hello, it's actually Mark's wife, Nicola Anderson.
Okay.
Hello. It's really interesting, the correlation between inflammatory disease. And I've been doing some work with periodontologists, Professor Ian Chappell from Birmingham University, suggesting that the increase in inflammatory disease in the mouth, the oral hygiene, has a massive impact on general health. That's health with the heart, the liver, the kidneys, everything. So it's quite interesting you saying that is it inflammatory disease that's increasing this calcification? And when you were saying this teenage boy, quite interesting as oral hygienist, seeing how poor teenagers' oral hygiene is and the increase in the hormones and how that has a massive impact on their general health. And when I qualified back in 1987, I saw very early in my career that my elderly patients with excellent oral hygiene had great health, yet my patients with poor oral hygiene were taking medication for everything, basically. So do you think there could be a risk factor with just poor oral hygiene and an increase in calcification in PXE, something as simple as that? It
is entirely possible. I don't know what triggers those possible flares of inflammation that we see in the French PXE patients. I would restrict it to the French PXE patient because it's the only place we've looked at and we've seen this. I can't believe it's restricted to the French PXE patient. And what we see in the mice, we don't quite see flare or evidence of flares of inflammation in the mice, but we see evidence of inflammation in the animals as well. Yeah, whatever contributes to this is unknown at this time. I have no idea how also ABC or the lack of ABCC6 drives this enhanced inflammation that seems to be occurring. Is there an external factor that drives this inflammation, like oral hygiene, as you mentioned? Yes, it's entirely possible. I can't say the mice have very good oral hygienes, but we generally don't talk about that too much. But yes, as you pointed out, oral hygienes has significant impact on health and cardiovascular disease and others. So yes, whatever drives it, I'm not sure. But is it possible that that oral health from teenagers contributing to PXE? Yes, I don't think it's a far-fetched idea. But there's no evidence one or the other.
Some people are more susceptible. I mean, I will have patients who I could scrape plaque off with a trowel, and yet they have no inflammatory disease, where I have other patients whose oral hygiene is actually fairly good.
Yes,
but you can see the inflammatory markers.
The point that you're bringing, that yes, susceptibility is different in between people, and perhaps people that are more susceptible to inflammation will have a more severe calcification than others. It could well be also an element that contributes to the viability of the symptoms in PXE patients.
Thank you.
Now we have Colleen and then Bob, and then a question in the chat that I'll read. Go ahead, Colleen.
First and foremost, thank you, Oliver, for your presentation today. I love the name Oliver, by the way. The character name in my book is Olive. Anyway, no, that's not what I'm talking about. I'm going to validate some of the things that you're saying, because I've been learning. I'm not a scientist, doctor, nurse, nothing. I have never been in the medical community in any way, shape, or form, nor into a student of it. I'm a student of myself. I have PXE. I was diagnosed at 19, had no clue what it was, never even researched it, just lived life, saved basically by the seat of my pants, like everybody, eating processed foods, doing nothing, no movement. Finally found out ATP is an energy that's within our blood. And I create my own now. And I have two methods. One is working out, doing my resistance in my muscles, and one is just breathing. Certain 30 quick, just breathing. That's my ATP energy increase. And it has healed so much of me. And there's even more, which Sharon can attest. I'm trying to heal my eyes right now without having injections. And I think I've done it.
I didn't quite understand absolutely everything. Sharon, can you help me?
I think just she's corroborating what you've said and wanted to offer that.
I think I heard ATP energy, and the rest was not super clear to me. So
I'm just really excited to say somebody who doesn't know anything about the medical community can now talk with the medical community, especially about ATP. And I think that expressing the cortisol from my system, which is the inflammation piece, helps a lot.
Great.
There's more.
Yep.
Thanks, Colleen. So from the chat, does TNAP function and inflammation interact?
It's a good question. I've been reading looking at it, and I think yes, there's a connection between TNAP function and inflammation. Yes, off the top of my head, I can't really give you any details, but I've seen papers that are making that connections, and it's important in inflammation. Yes.
Someone else writes, I see that the thymus gland can be affected by PXE. I have not heard that. And that, and it also myasthenia gravis. Is there a link between PXE and myasthenia?
I haven't heard that the thymus is involved. Myasthenia gravis, I have to say that I'm not sure what this is, so I can't really speak in an educated fashion on this. The thymus is part of the the immune systems, and maybe, but so far we have not seen evidence. But this entire story that I just described is really show you anything. It's not because we have not seen it, then it's not there. So I can't really give you a good answer on this question.
Okay, Artie, your hand is up.
Hi there. So first of all, thank you for this wonderful presentation. Second, my question is, because we're bringing up inflammation, PXE, and how they seem to be kind of interlinked in some way, is it possible that things that may be linked to inflammation, like fibromyalgia, for example, could just actually be PXE, but they're mistaken as something else?
I can't really say for sure. There are a number of ways to diagnose PXE. Misdiagnosis is possible. But I can't say for sure, really. That's another area where I'm a molecular biologist, I'm a PXE expert, and I'm not an immunologist by any means. So I'm going to have to dig myself into inflammation, and I need to educate myself further in this. So I'm sorry I can't bring you any better answer than that, but I don't really know for sure. And there is a question in the chat that says, what's next? What's next is to figure out how inflammation comes about. What is the molecular driver? What is the trigger of inflammation? And how this is regulated? What role ABCC6 has to play into this? What immune cells? So where is this T cells, B cells? We know it's not macrophage by default because there's no ABCC6 expression in those cells. So we're trying to hone on really the next level is going to be figuring out how this process happens. What is the molecular mechanism behind it? And once we find the molecular mechanism behind it, then we can think about a possible intervention. And whether this would be a useful intervention, I can't say, but this is at least what we're going to be working for.
Great. All right. Well, thank you so much, Olivier, for sharing your knowledge. I want to remind people that we're going to hear from a lot of speakers. Lots of their evidence will be contradictory from one another because we are bringing you cutting edge research. And a lot of this has been done in animals, which translates sometimes beautifully to humans and other times requires a great deal of work to transfer to a human. I've also gotten quite a lot of questions about, so will there be a treatment the latter half of this year? The answer to that is absolutely no. We will not have a treatment for PXE this year. Even the ones that started 20 years ago still need a great deal more work. The saying in drug development is it takes about 20 years, $1 billion, and a 95% failure rate to get to a therapy. So we've got to do 100 shots on goal to get 5 of them in the net. And we need $100 billion to do that. Obviously, we're looking for lots of ways to make that possible. But work like Olivier is doing contributes a great deal to the body of knowledge. And we'll continue to say at PXE International, the very best thing you can do is lead a healthy life, eat well, make sure you exercise, make sure you take care of yourself, because whatever is going to come out at the end, we're fortunate, and I know that's a terrible word to use, that this is a slowly progressing disease, which means that if we can mitigate it, we can interrupt it somewhere along the path, and we have time to do that. Unlike these diseases that kill kids at 5 years old, we have some time to have you make a difference in your lives the way that you have been working. So we appreciate, Olivier, everything you've contributed from the very beginning until today and beyond.
Yeah. Don't go out and take ibuprofen. That's probably not a good solution at this time. As I said, we don't know what drives this whole phenomenon. What Sharon had mentioned, I just wanted to say briefly, is very important when I conduct research is to do this in human and mice. And we look at what's actually crossing path and what's common between the two because of what Sharon mentioned. What we see in the mice is not necessarily translated immediately to humans, but it's a really important model to work with animals. But confirming what's happening with human samples and so your contribution to research is very important to further the field. So I just want to say a quick hi to Anna, Susan, and Viola are just joining that.
Yes, and collaborators for a very long time as well. Great. Well, thank you, everyone. Join us for our next one, which I should probably be able to announce to you, but I can't, and you'll be getting an email from us about who you can see next. And this will be in our library of talks shortly, sometime this week. All right, have a good evening, afternoon, morning, wherever you are.
Thank you.